other thing here. Okay. All right. So welcome everyone to our Friday Hack Hours. We are doing another session on boosted regression trees, and these next two sessions are going to focus on, you'll learn more about them and how they're used, but it's kind of learning by seeing some more simple applications. And I'm really happy today to have Ryan Vandermeulen, who taught the Fish Pace Hack Week. Gosh, been over a month now. It's the end of January. And we're going to see a tutorial that we taught during that Hack Week. This is using PACE hyperspectral ocean color data and some SEAMAP survey data on fish abundance to just kind of play around with, making like a little prediction model using PACE data. And today you're going to see more about how to do the matchup part. So you've got some lat long time data and you want to match that up to some remote sensing data. So I jumped right over that part last week, and because I knew this week we'd get a little more into that. So you're going to see a specific example of how you can do that, and then Ryan will jump into using a boosted regression tree to make these predictions. All right, so I have a page up here. I will put the chat in. I'll put this link in the chat. If I can find it somewhere. Come on. Yeah. All right, there we go. So we are going to be using this session, next session, we'll be using the NASA PACE data. It's a new mission to get hyperspectral ocean color data. You'll learn a little bit about that. And if you want to follow along by actually doing it yourself, you can do that either in our NOAA Jupyter Hub, or we also have another one, our NMFS Jupyter Hub. That one has a persistent home directory. Reach out to me if you can't get on that. You're welcome to. The NOAA one does not have a persistent home directory, but anyone from NOAA can just log in. And then you can always use Colab on these. So, but because we're using the NASA Earth data, you will, if you want to play along and just follow along, you will need to have a username and password. And I put this in the chat. It's free. You just sign up. Just make sure that you have that username and password written down because you're going to be asked for it in the tutorial. Okay. And then, so here's our, the one we're going to be doing today. And when you click on that, you'll see this. And so there's a few different ways you can open that up. You can download it if you're set up for Python, or you can open in Colab, or you can open in one of our hubs. All right, I'm going to stop there and hand it over to you, Ryan.
Oh, thanks, Eli. Appreciate it. I'm super glad to be back at the Hack Hour here. It's especially to talk about something I get super excited about. All right, and just to make sure we're all on the same page here, here we go. I'm going to
I can't hear you, Ryan. Was that only me?
I know why. Okay. I was trying to share I'll have to swing back and forth, but we'll see. Okay. Anyway, so I just wanted to get everyone kind of up to speed. We can give a little 2.5 minute quick overview of the nature of the data we're working with today. So we're focused on ocean color satellite data. You don't need to be too familiar with the science of it for today's exercise. Just know that our ocean color satellites in orbit are really nothing more than fancy eyeballs, looking down at us from space. So our eyeballs can, detect roughly 1 million different colors or so, but fundamentally we're only integrating, the intensity of 3 basic colors: red, green, and blue. And there are a handful of folks with tetrachromacy in the world, which is this genetic condition enabling the perception of 100 million colors or something crazy, just due to the presence of having a 4th cone in the retina. A nifty little rabbit hole if you ever want to go down it. But I think I heard somewhere it's thought that maybe 12% of women globally have the genetic predisposition for it. So anyway, besides the point, 3 cones based on the relative intensity of the amount of blue light, green light, and red light coming back to our eyes. Our mind integrates this signal and basically sends us a single answer back: I see olive green, or I see brown. So I like to think of this like when a piano or a guitar kind of strikes a chord. And I'm going to have to switch back and forth here and go off of audio because Zoom is being fun here, but just give me 1 second. So if we translate each of these light intensities into a pitch and kind of play them all together, it might sound something like this. Now, the satellites see a lot finer color scales than we do, right? So that when they see color, it has a bit more nuance to it, and it's going to sound a little different, such as. Okay. All right. There should be enough of the going back and forth. But because we're separating out, more wavelengths of data, is really, and I hope that played. I have no idea if it did. It should have. It gives us the opportunity to sort of fingerprint the ocean based on its color intensity. So this fingerprint is just a really, the basis for calculating cool things like chlorophyll A from space, which I'm sure most of you have seen images of. Okay. Rewinding back to February 2024, NASA made tetrachromacy look like child's play with the launch of PACE, which is the Plankton Aerosol Cloud and Ocean Ecosystem Mission. So for the last 27, I think going on 28 years, going all the way back to the launch of a CWIFS satellite in 1997, our view of ocean color has been based on measurements of only 5 to 10 sort of discrete spectral bands or color bands. So it's been incredibly useful, and it's gotten us a long way. You get things like chlorophyll A retrievals that are, like I said, built on that foundation. But that paradigm shift, there was a shift in that kind of when PACE was launched, because what it brings to the table is having this hyperspectral capability. So instead of sampling the ocean at a handful of color bands, we're now filling in all of these gaps and measuring this near continuous color spectrum, giving us access to a lot more specific information that we really haven't had access to in the past decade. So we call this hyperspectral resolution. This comparison just shows how dramatic that jump is when you put the PACE sensitivity next to heritage sensors like MODIS or VIIRS. So PACE is really meant to provide this continuous, uninterrupted sampling of the visible spectrum, and even get us into the ultraviolet and near infrared portions as well. What this means practically is that if something's modifying the light of the ocean and it's happening at a large enough scale, PACE gives us a chance to see it. So I'll wrap up here and just say that, this is important because there's a lot of information contained in the ocean color signal and whether or not biomass or productivity is high or low. Two regions could have very similar, biomass of phytoplankton in the ocean and still represent completely different ecosystems. So, just like flowers on land come in different shapes and sizes and occupy different ecological niches, the same is true for phytoplankton in the ocean. They all contain chlorophyll A, but they also contain other pigments, other chlorophylls, carotenoids, and things like that. So the presence or absence of these pigments can be associated with different phytoplankton groups. And having access to these sort of subtle spectral features may help us distinguish broad community types and separate out detrital material from sort of the living components. There's also some signal in the way that cells scatter light as well. The point is that not necessarily that we can definitively separate out all these processes, but with having this hyperspectral data from PACE, we now have kind of a much better opportunity to really dig deeper into these optical signals, and which could really reveal some critical insights into ecosystem function and vulnerability, especially where when we're talking the bottom of the food chain here, where the size and sort of energy quality really makes a difference as it cascades forward. So, all right, that's my ended up being more than 2.5 minute spiel there, but. All right, so let's jump into the and the screen here. All right. All right, then. So you guys see the screen all right? All right, cool. So, yeah, we're a lot of fishery science is kind of living in point space here. So we're collecting data at very specific locations and times, and we get trawl tows and plankton nets, CTD casts, tag locations, what have you. There's a lot of sort of information, rich. There's a lot of information sort of contained in that spatial data, but they don't sort of tell the whole story on their own. A lot of times we want to know as well, like what's happening in the environment, where those organisms were experiencing at the time that we observed them, which was a really powerful aspect of having access to satellite data. This is where it's satellite data can be really useful, but could also get a little messy. So pulling a single pixel from a satellite image is usually pretty easy, but once you start dealing with a lot of points spread across different days and maybe even years, the process can get kind of slow and painful if you're doing it by hand, essentially. So in this tutorial, we're going to build up a workflow that helps automate that matching step, lining up fisheries observations with the right satellite data in space and time. So we'll take it one piece at a time so the logic kind of stays clear and reusable here. And also, instead of having this in sort of a hypothetical space, we wanted to fiddle around with some real fisheries data. So we're going to be using data collected by one of the NOAA, one of our biannual surveys collected in the Gulf of Mexico from SEAMAP. If you haven't worked with this data, it's super cool. We've got species composition, size, abundance, environmental measurements, all focused along the U.S. Gulf Coast. So it's a cool sandbox for experimenting with all these kind of integrations. So by the end of the tutorial, there's a few things I hope you can come away comfortable with. One is your run-of-the-mill pixel extraction from a satellite image. Then we want to import some real ship survey data and kind of visualize those sampling locations directly on top of a satellite image. Then we'll extract that data and all the appropriate locations and dates for making a bunch of observations, for having multiple observations at once and not just a single point. And this is all building up to being able to just get the right inputs to get into a boosted regression tree, all the sort of dirty work that goes to get to that point. And so we use that combined data set to build just a really simple proof of concept model that just relates fish abundance to satellite-derived variables. Nothing fancy here. The goal here is just kind of showcase a workflow, essentially. So, all right. Let's start with a simple exercise here. We're going to extract pace reflectance data from a single defined location on a single day. Feel free to change the date and location below. But for this tutorial, I did pull an example that was relatively cloud-free, so we could get results here. And so let's go ahead and define that, and then we're going to use our friend Cart. We figured out how this is pronounced, Eli. Is it Cartopy? I always wanted to say Cartopy, but the Cartopy toolbox here, library here to help us with mapping. So the first thing, without any satellite data, we're just going to take a look at where it is that we're sampling here out in the Chandelier Islands. Really cool estuarine region here. All right, so let's grab a PACE image here using this Earth axis tool that Eli mentioned. You need login, and I hope my access hasn't expired. I think we're good. But we basically queue NASA for data based off of we can deliver a short name of, and it's kind of a whole another aspect of finding the data. But long story short, we're queuing a specific product, remote sensing reflectance, which is that raw color information that we were looking at. We're defining what type of data. This is level 3 mapped data, which means that it's been sort of gridded onto a common map, and not sort of the way a satellite might see it, where it might look a little warped as it's sort of going along a globe in orbit. And we're grabbing things with data from the PACE observatory, and the OCI is the Ocean Color Instrument. So this is all sort of defined in the short name. We defined our time scale up here for the day that we wanted, and then we're grabbing just a daily image at 4-kilometer resolution. This is all sort of contained in the file name here. All right, so we're just going to just open up the data set here. All right, so we predefined the latitude and longitude here. So yeah, here we're opening it up here with a file with latitude, longitude. There's 172 wavelengths. So each wavelength is just a color band of information. So every single pixel in an image is going to be one of those sort of full color curves that you saw. This is just looking at the structure of our data. So we had predefined latitude and longitude at the top. So we're going to use this .SEL here just to grab the reflectance at that given point, passing it this nearest neighbor command, just to give you an idea of what. So this is what a single pixel extraction looks like. All this color information. If you're not familiar with sort of spectral color bands, wavelengths in the 400 range are about at the limit of what we can see in the blue to about 700 in the red. So here's our green. What this is telling you, we're looking at the reflectance of the ocean. So what this is basically telling you is high reflectance here along the greenish, sort of yellowish portion. We're looking at probably some olive colored soup here, essentially. Further down into here is into the ultraviolet, which we can't really detect, but still has an important component in ecology. This also tells us that there's a lot of things that you can tell just by looking, not just the color of the water, but having this really low ultraviolet reflectance tells us there's a lot of dissolved organic matter in the water. UV light gets absorbed like nobody's business. It's the best sunscreen when you have a lot of, if you're sitting outside the Mississippi River plume and there's a lot of sort of tannins and dissolved organic matter, it really loves to eat up that UV light. So there's also a lot of phytoplankton in here. You see this little bump here. This is centered around sort of 680 nanometers. This is where we're measuring fluorescence line height. So phytoplankton are absorbing sunlight to photosynthesize. Essentially, not all that energy is getting used. Some of it gets re-emitted as sort of a faint red glow. So it's basically excess light that didn't get used, and it's giving us a direct. This little bump here is just giving us a direct optical signal that there's living stuff in the water, and it is phytoplankton. Okay, so let's throw a few more points at it. We're just going to build a real simple loop here, and near instantaneously as we go through here, we get a return here. It's pretty straightforward, and it takes little code to do, but let's just. Adding a simple addition somehow just complicates things a lot more. If we want to do the same thing but over multiple days, and talking a lot of days, and if you're working with a long-term satellite data set like MODIS, and you've got 20 years of fisheries data you're trying to match up, you might be extracting thousands of points from hundreds of days. So now we're just going to kind of take this into a real world scenario here. As I mentioned up front, we're going to work with some real-time ship-based survey data coming from SEAMAP, specifically from 2024 onward. I kind of took the liberty of pre-extracting some data into a CSV just to make it a little more user-friendly. So we're not pulling things in multiple files, but the idea here is simple. For each survey station, go ahead and load this up here. So for each survey station, we want to reach into the satellite data record and pull out this PACE remote sensing reflectance values at the same locations and dates. So this kind of just gives us a clean way to pair our point-based biological observations with spatially continuous satellite data. And here's all the things that we're able to pull out of this. Includes latitude, longitude, sampling date, and a handful of in situ measurements that were collected during the survey: temperature, salinity, and oxygen, chlorophyll from fluorometry on board, turbidity, Secchi depth, all sorts of cool stuff. For this, we also wanted to pull some sort of fisheries response variable. In this case, we're going to use tote live. Here we go. Our total live. This represents the total wet weight of everything that was caught in tow: fish, crustaceans, the whole works. Whatever came up in the net. It's not the most nuanced fisheries metric. There's a whole bunch of cool species data and everything, but it's right now you just kind of want to demonstrate how this kind of satellite survey matchup works. And the nice part about this SEAMAP data is if you want to go deeper, there's a lot more under the hood here that you can get into. And if you want to adapt this for your own work, I should mention as well, you don't need SEAMAP data specifically. This is written in such a way that you should be able to ingest any CSV file that has named latitude, longitude, and date. If you want it to really work seamlessly, name it LOC_date, and it should read in seamlessly. So as long as the date is sort of formatted in this way, should plug in. All right, so we use pandas to read in these CSV files, which it's just heaven sent. It's really useful. I don't bother with Excel files. It's worth it, the trouble to go through and and convert, multiple tabbed Excel files into CSV, because it works so seamlessly in Python. So, all right. Yeah, we're pulling. We named track DF. This is our pandas reading in the CSV. This is live here, so we're just getting, all of the. I think this is in kilograms of total weight, essentially. All right, we're also going to pull the, on any given day, they sampled sort of more than one station, right? It's not just one station per day. So if we're not careful, when we set this up later, we could end up pinging NASA for every single 594 lines that we have in this spreadsheet here. Okay, and we don't want to do that. For every single date, we want to retain that date and say, and we want to do this quick thing where we extract multiple points from a single day, instead of having to go and ask NASA for permission to access a day over and over again, to help make things a little efficient here. So we're going to pull all the unique dates associated with this, and we have these 61 dates total. Okay, so let's just take a moment to kind of unpack what's happening in some of this. We're just going to plot up what all the locations of this on top of one wavelength of reflectance from PACE. This might be, if this, you guys have kind of gone over all of this before. I hope it's not too elemental, but just I feel like a few things that are just worth mentioning are using this slice function. It's always good to hear things repeated as well, but it's not just a spatial trim. We're not changing the data. We're just limiting sort of the map extent, so we're not plotting the entire globe here. And then we pull out the 443 nanometer wavelength, again using this nearest nearest component here for just in case it happens to be 442 or something like, and it's like 442 nanometers. So you can grab the closest wavelength. This is a common one used, as it's the peak of phytoplankton absorption and for chlorophyll, for instance. So anyhow, let's see. Moving on from this, you can just see we have a plethora of great data points that extend over the course. This is just one day, but you can just see from 2024 onward how much data has covered the shelf of the Gulf here. All right, so getting to extracting multiple points here. I don't want to dive too deep into this, but just in case you're interested in, if you have some familiarity with dealing with satellite data, it's often provided in these different bins at 8-day or monthly averages or daily data, and these composites can really, depending on what your question is, it can really make a difference. What sort of data you want to pull, and the right answer really kind of depends on your question. If you're in terms of fisheries, if you're looking at really broad habitat use for a really highly mobile species, grabbing the nearest 8-day weekly average is probably fine. Most of the features they're responding to probably persist on sort of weekly scales. If they're interacting with offshore eddies and things like that, or frontal boundaries. But if you're working with things like plankton toads or shelf species that are kind of tightly coupled to more short-term processes or tidal influence, daily data might make more sense. If you're looking at climate signals over the course of 20 years, you might want to look at monthly data, so you can kind of detrend it and things like that. The trade-off is cloud cover. Daily imagery drops down your sample size really fast. In our case, switching from an 8-day to 1-day composites takes us from about 500 matchups out of the 530 some odd points that we have down to under 200. So if your science question can tolerate a little bit of sort of temporal smoothing, longer composites can really help with coverage. Just be really clear about the assumptions that you're making. In this case, we're not tracking tuna across the Pacific. This is trawl data from the continental shelf. Things like river plumes and mixing can change really quickly. So for that reason, in this particular instance, I might keep the temporal window tighter in this case. But again, depending on your question, you could be working with this exact same data set and maybe want to change your compositing. Okay, so at the high level, what we're going to do next is kind of scale up this single matchup to many matchups in a way that's memory safe and efficient. So for every unique sampling day in our CSV, and before you run this, I'm probably not going to run this here because it takes about 5 minutes to go through, and we are going to grab the export of this file just down the way here, so you don't necessarily have to run this if you don't like. But for every unique sampling date here, in our CSV, we're going to query NASA for the corresponding daily level 3 PACE file. And once we have that file, we're going to loop through all that survey data collected on that day and extract the nearest satellite pixel for each one. And rather than writing to disk over and over, we're going to store everything temporarily in a dictionary, and then collect those dictionaries in a list, and then append everything back onto the CSV in one shot at the end. It just kind of keeps things fast and tidy. I know 5 minutes seems relatively slow, but we are going through quite a few data points here, and the real bottleneck is having to ask NASA over and over again for access to this information. So one important sort of implementation detail here is memory management, and it took some back and forth and a lot of experimenting to try to get this right. And there's still probably ways to optimize this even better, I'm sure. But we only ever open one satellite data file at a time. The Earth Access Open call here streams the data, so nothing gets fully loaded into memory unless we actually call it with Xarray. On top of that, there's a with statement here that makes sure that each data set is closed immediately after we're done with it. So once that block ends, the memory is released. Helps you keep your kernel alive, especially if you're working in the cloud. So this day by day approach here, this is sort of the bottleneck here. If you have a lot of local memory, it can be faster to just download everything first and then just process it all at once. It will be much quicker. But we're choosing stability and sort of portability over raw speed here. And like I said, it only takes a few minutes here, but for the purposes of the tutorial, feel free to skip over it since we provided the output. Let's see, anything else I want to mention about this? Another modification of this is the 3x3. In satellite workflows, we're instead of just grabbing a single pixel, we might sort of average over a small neighborhood, 3x3, 5x5 pixels. It can help smooth out the noise or reduce some of the impacts of clouds or bad pixels, and it kind of gives you a sense of local variability as well. Even though these sort of level of products have already been binned to sort of a coarser resolution, the same idea still applies. For habitat questions, a little averaging might better reflect the environment a fish actually experiences rather than the value at one sort of exact grid cell, because they have probably been traveling all around, throughout that day. So this just shows a very, very small sort of modification of that, in which we're just computing a 3x3 mean around a survey location. It's just a really small tweak, and the action sort of all happens in this little section here. When you use the Xarray select the nearest method, it it gives you the data value at the closest coordinate, but doesn't tell you the actual index position of the array. So if you want to grab a block of pixels around that point, you need the indices to kind of slice the array. So which is why we use this this absolute value of the latitudes, the latitude values and longitude values that are in our spreadsheet, minus the latitude center here. It's just giving us a numeric index directly, which makes it just easier to define that 3x3 window around the point. So essentially, we're indexing the location of the nearest lat long we're interested in, and then create a small neighborhood around that point, and then take the mean. All the other logic stays the same. One other thing that was added as well, we're extracting the way some of the PACE data works. The reflectance data is more three-dimensional. Each data point has a whole stack of all this wavelength color information. You might just want a single product like chlorophyll, so just a two-dimensional data set, and it's not in 3 dimensions. Again, just some pretty. I think all the magic happens here. It's just a slight modification, but just wanted to include it in these scripts so that way you have for various different ways of doing this, you have access to whatever you might want to extract. Okay, so we've matched up all of our survey points to a satellite spectrum. Just take a quick look at the data here. We're going to focus on this total live variable in the SEAMAP data again, which represents that total catch in kilograms. And for for something like habitat modeling, we're not necessarily going to expect a super tight correlation between remote sensing, reflectance, this color that we're looking at, and total catch. But it's useful to explore how PACE data might help. So, all right, first we'll open up our CSV and just get a sense of the average reflectance shapes across different fish biomass levels. So if we print out the minimum and maximum of the data here, we see that it expands 3 orders of magnitude, and it's biological data. So this immediately tips you off that we're probably going to want to work in log space here if we're dealing with multiple orders of magnitude and biology, right? So if you take the log of this data, it normalizes the distribution. So when you apply, parametric statistics down the road, you don't have these extreme outliers that are driving your correlations. And so what we want to do below is essentially we'll transform the data by taking the log 10, and then since it's more evenly spaced now, more normal distribution, we'll evenly divide it into 10 bins. And within each of these bins, we'll take the average of all the reflectance data just to see what that looks like. So in general, we see higher catch in sort of, remember, the way to interpret this with these reflectance values, here's our wavelength here. So red light, blue light, and more green light. So when you see higher reflectance values and the sort of shift in the peak more in the green area, you're seeing greener waters, but we're seeing more teal waters kind of here, sort of almost brown olive green shift and down here. But so in general, we're seeing higher catch essentially. Our brighter colors here are higher bins of catch in more greener, presumably phytoplankton-rich waters, and then we're getting lower catch in bluer waters, which is not too surprising. One interesting exception here, though, is this low biomass bin. Our first bin here is just not much biomass, but it's really green shifted, but the overall reflectance itself is much lower. It's just a lot different from what we're seeing here. This likely points to a lot of strongly absorbing materials. If we have like a lot of CDOM, coming out from river runoff, there's a lot of light being absorbed and just not letting it get to depth, right? If this was the case, and we are seeing some sort of kind of CDOM or salinity like marker here, it should also show up as lower salinity. It doesn't really fully explain the biomass yet. So let's it might be helpful to look at some of the other environmental parameters in the same biomass bins. We are purposefully getting our hands really dirty with the data here, really exploring sort of what this is before we sort of put it into the black box that is machine learning. Really want to kind of be familiar with what it is that we're looking at here. So this is leading up to what we're trying to get at here. So yeah, let's see. Taking a look at some of the other environmental parameters here, we can pull out surface temperature, surface salinity. The Sal max is the maximum depth salinity. So this is bottom salinity, bottom oxygen as well. And just kind of this aha moment here in our bin that corresponds to what we're looking at here. This 0.43 to 0.94 bin. There's a really big difference between the surface salinity, so relatively low, and the bottom salinity here. So we have this sort of freshwater lens stratifying the water column. We have high nutrients, low light at the bottom, not really getting through, a lot of absorption happening. Essentially, the water is getting trapped, oxygen gets depleted by microbial respiration, and bam, we've got hypoxic waters, and our concentration of oxygen is below 1, borderline, anoxic here. So it's no wonder why we're not seeing any fish here. So it's slightly a bit of a off topic here, but the takeaway is that just by looking at some of these reflectance patterns, even if we're just kind of summarizing, into these bins, you could just look at this and kind of flag areas with unusual environmental conditions. We looked at the data and it's like, wow, something kind of sticks out there. So this is where it's helpful if you sort of have an understanding of some of these aspects of what types of materials in the water absorb what types of light. It can give you a real leg up relative to looking at something just like chlorophyll, which probably would not have tipped you off to this at all. If you're just spinning it by chlorophyll, I don't think it would have really popped out. So, okay. So this is a fun thing to do now because we have this nature of hyperspectral data and help us figure out what to sort of feed into a boosted regression tree next. There's a couple things we could do. We could just, if we're trying to build a model, one school of thought is we could just put all the reflectance data, throw all the reflectance data at it. Boosted regression trees do a pretty good job of dealing with, cross correlations and things like this. But we're gonna take a different approach here, and again, just diving into the data and trying to get an idea of what's happening. Let's just try to look at the hyperspectral patterns and see if there's, with all this hyperspectral data, let's see if we could pull something out that might be useful. So we're gonna do this with a correlation matrix. Having all this hyperspectral data, we can start kind of teasing out which spectral features might relate to fish catch, even if we don't expect a strong relationship. So we're going to use this. So here we're just extracting all the reflectance between the wavelengths that we can see from blue to red. Outside of this range, the data gets a little noisy. So we'll just keep it within the range of data that we know is kind of higher quality. So we're going to use Tote Live. This is our fish catch as our target, and then we're going to compute a simple two-band index here, sort of normalized difference index for every single possible pair of bands that we have. So we could use different two-band combinations, a ratio, or something raised to the exponent. We're just using an NDI, it's a normalized difference index. It's easy and kind of well behaved. So for each sort of band pair, we'll calculate the normalized difference index, and then we'll correlate it with our log transformed fish biomass. And then plotting all of those correlations as a heat map kind of lets us quickly see which band combinations look most promising. And we have over 100 bands from PACE, so we can explore spectral relationships that maybe haven't been tested before. All right, so we're going to give that a spin. Might take a few seconds as we're doing, again, all these, every single sort of band combination. We're calculating this normalized difference index against them, and we get this kind of cool heat map here. A few quick notes on the code here. This double loop here kind of computes just every possible two-band NDI and then correlates it with our target variable. Let's see, we used anything else about this? We end up masking out. Where is that here? Yeah, here we're masking out the sort of this white portion of the triangle here. We don't have to do that, but it's going to be symmetric, so it just kind of helps make it a little prettier. And here's the point which we're actually calculating our coefficient, which is the value that we're using as the basis for the heat map here. Everything else here is all just formatting the plot to look nicer. So basically, the deeper red and deeper blue areas are just showing which wavelength pairs show the tightest linear relationship between NDI and the log transformed total biomass. So it's a bit qualitative. So we can use this matrix, this data matrix that we have to pull out the band pairs that sort of show the top 5 highest correlations. And because we're hyperspectral with a lot of cross correlations, it's no surprise that the top 5 highest correlations are essentially varying versions of the same band combo, this 400 to sort of 650-ish. We can expand this to find others, and you can list out 100 and then wait for the next sort of ones that are separated, or we could write some fancy code as well that tries to separate this out. Oh, and I see we're getting 50 minutes. Let me move on here. Sorry there. Thank you.
Give you a quick time check there.
Yeah, I appreciate it. All right. Okay, so what we're going to do is put this into, we chose this 2-band pair. We're going to put them in and calculate this NDI ratio and these other environmental variables, and set them up to get into a boosted regression tree here, pulling in our all of our libraries. So here's our predictors that we're going to use. You'll notice we're now down to 97 samples because we're matching pace data with all the areas where we had ship data as well. So not a huge amount, but it gets us where we need to be. All right, so we're just doing our setup here. We have our training and test data set. Let's see. We're going to split this up into an 80-20 sort of test and train. 80% for training, 20% for testing, and then you set this sort of random seed for reproducibility, which you may or may not want to do, but for the purpose of if you want sort of the same results when you do this over and over again, it's very helpful for that. So we're going to fit the boosted regression tree. It's basically just builds a lot of small decision trees sequentially, and each kind of learning from the errors of the previous. We give it sort of a relatively small learning rate and subsampling to kind of help reduce overfitting. We don't have a huge data set here, so we want to try to minimize the amount of overfitting. And then once trained, we're going to pull out some of this the 20% held out test data, and evaluate some of the performance using R squared, RMSE, and mean absolute percent error here. So 0.709, not too bad. R squared 0.404. Yeah, like I said, we weren't expecting something super magical here, but that's nothing to sneeze at. That's kind of what we're seeing in our correlations that we were seeing beforehand. So, all right. So to visualize the results, we have 2 plots we can make: a scatter plot of observed versus predicted values, and then basically a star polar plot here, which shows the feature importance, which is a really cool aspect of boosted regression trees here that you can pull out and visualize. So in this model, surface temperature had sort of the biggest influence, but the pace normalized difference index that we calculated was very close second. So this model actually kind of captured a pretty nice directional trend. But remember, we only have 20 points here in the test set, so not a super robust predictor. But interestingly though, if you go back and remove the pace data, you get a lot more observations because the cloud cover is an issue. We're not relying on the satellite data. But the model's predictive relationship weakens pretty substantially, just showing that even a small amount of kind of having this hyperspectral information can add some value even with those limited samples here. So yeah, this is where the science sort of begins. This is meant as more of a workflow tutorial, helping match up sort of observations to environmental data and exploring relationships. Again, a really important aspect. Getting familiar with your data, exploring the relationships, and then sort of having those experiments beforehand to lead you into what you kind of want to put into your predictive model. So we didn't just throw everything under the kitchen sink at it. So I'll leave it there so we have some time for questions. But I got too talky on a Friday afternoon here.
So I'll kick it off with some questions. So it looks like the points are pretty clustered around the shelf, perhaps. And I can't remember if you included bathymetry
As one of the predictors.
Yeah, bathymetry or distance from coast. Because I'm thinking, like, if you were to take this kind of approach and what you wanted to do was create like maps of potential abundance. Um, yeah, I guess I'm wondering what other parameters you think you need to like put into that.
Yeah, that's a great question. A lot of things that might come out in a uh, species distribution model. Bottom
Yeah.
temperature is often a huge predictor. Depth itself, that and certain aspects of the sampling time of year as well. We threw everything from 2024 onward, but you're going to have different distributions in spring relative to winter, and so yeah, kind of knowing what it is you're looking for as well. Even things like I've seen, grain size and sediment type. If you have that sort of information associated with it, especially for these are trawl surveys, so we're looking at benthic critters, and it's and I know too it's not lost on us. We're talking about satellite data. We're talking about a near surface signal, right? And to some degree, there might be some aspects of visibility and light availability that critters could be responding to at the sea floor, which could be really important and be informed by satellite data. But a lot of times what we're looking at with satellite data as well is the manifestation of oceanographic features that it might not be necessarily the taste of chlorophyll in their mouth, but you're seeing through this data a frontal boundary. You're seeing a river plume, coming out of the Mississippi River. You're seeing oceanographic processes being manifested. Sea surface temperature is a really good one, but if you're working in the Gulf in the summertime, you will know that the entire thing turns isothermic 28 degrees C or whatever it is, and the entire Gulf just looks like one big red blob, and there's not a lot of structure there. The color aspect is nice because it helps you identify some of those features. You can still pick up the loop current eddy and different features coming out of the estuary year round. And then you can also bring in other things like altimetry that aren't necessarily things you can see, but just sea surface height.
Mm-hmm. Looks like there's a question in the chat. Can you get uncertainty bands around your predictions from boosted regression trees?
Uncertainty bands around your predictions. Let's see. One way I can think of, and you might have better answers. One way I can think of doing this immediately off the top of my head is to kind of pull a instead of having your static. Where do we set that? Am I still screen sharing? Yeah, our random state, so that it's reproducible. what we might want to do is non-random to make it random, and then run that say 1,000 times and sort of take the average of that and a standard deviation, basically doing kind of a Monte Carlo type of approach and seeing if you're getting, sort of what the what do you call it, the ensemble of your answers are. If you're taking a different 80% and 20%, and sampling it in multiple different ways. That's one way I could think of getting a good stab at uncertainty. Are there other? Do you know of any other sort of through the
Yeah, the approaches I would think of are some kind of bootstrapping where you're holding, so you gotta have enough data to hold out. And once you can hold out data, then you're able to, well, you kind of showed it if you scroll down and you show that, yeah, down, 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 your performance right there. So that is a type of uncertainty. So you could um, kind of derive from this an estimate of how far off your prediction, the red line, is from what you're observing. But if you'd want a lot of points, so you could get um, good estimates of that. So here it is. So what he's showing is a two-dimensional observed tote live predicted. But imagine you had a lot more test points. So imagine that. You could add some other dimensions. So you could say, hey, when observed tote live is this and the temperature is this, how far off are my predictions from the observed? So you can kind of, if you had enough test points, you could sort of map out what your uncertainty is.
Yeah, it's helpful ideally to have a completely independent test data set to sort of map out some of your uncertainties and test it out on as well.
Mm-hmm.
Bootstrapping is a perfectly valid thing to do. It's just it's not always as ideal as having an independent data set, but a lot of times because you're essentially training your data and then testing it out on the same data set, right? So we're trying to like randomize that as much as possible so it's not introducing that bias. If you have a representative enough data set, a global data set that's covering all representations of every single instance that could possibly happen, then that's perfect. If you're regional and you're bootstrapping and then you try to take this to another coast, you might
Mm-hmm.
that's kind of where you might run into
Yeah.
some problems.
There might be a large number approximation. I haven't used them enough and to maybe really a simple answer. There may just be some analytical approximator, and you're looking at your fit, and then your test and the training data that you're fitting to, and you're making some assumptions, and you can estimate the sampling variance or something like that.
Yeah, yeah.
All right. Well, it's 1 o'clock, folks. Great to see you. Have a great weekend. And we'll be back next week. We're going to see another application, a little more going more into less about the data munging and sort of prepping the data and more about like interpreting the results, looking at feature importance, that kind of thing. And we'll be seeing another example using fisheries survey data. This will be using silverhake data and using boosted regression trees to come up with predictions again. All right. Thanks, Ryan.
Thanks.
Bye.
Bye, everyone.